First of the 3 steps, and this is essay number 1, is the first 2 steps, and I've listed them up here like an outline. Blood vessel spasm. Tunica media in response to an injury is going to go through a pretty severe vasoconstriction, but it can only last about a half hour. But that will help other actions limit blood loss if the overall diameter of the rib vessel is a lot less. Second is the platelet plug formation. Now notice the last words in this paragraph up on the top. This is not the blood clot. This is like spackle against the torn blood vessel wall. Here we have a positive feedback loop, and the name of this positive feedback loop is the platelet release reaction. One of the things that we talked about with platelets, and this goes all the way to slide 29. If you look at this picture, platelets normally circulating in the blood look round and passive. When they become activated, and this is what it's talking about on slide 29, they will become chemically activated by trauma, and then they look all like the platelets on the bottom left, all pissed off. Those are activated platelets. And that's what actually is forming in the torn wall that's shown there also on slide 30. But on slide 32, we're going to talk about what happens when they become activated. The way they do it is they bind to something that is negatively charged. And when you tear open a blood vessel wall, the connective tissue behind the endothelium will expose collagen and protein. When you put that out in fluid, now this is exposed to plasma. When you put proteins in fluids, they tend to go negative. So there's a negative charge on this collagen, and the platelets bind to negatively charged collagen, and that's what pisses them off. And they look all spiky and angry. While they're becoming spiky and angry looking, they are releasing granules of ADP and a prostaglandin, which is derived from fatty acids, called thromboxane A2, or TA2 for short. This activated platelets, by releasing this, triggers the positive feedback loop because other platelets are attracted to ADP and TA2. They come to where the blood vessel wall is torn. They get pissed off looking. They release ADP and TA2. That attracts more platelets. They get pissed off. They release ADP and TA2. That attracts more platelets. They come, lop onto the other ones, become pissed off looking. They release it. Do you see the positive feedback loop? It just goes and goes and goes. Also, the platelets are releasing serotonin, which will help to maintain the blood vessel spasm for more than 30 minutes. Again, though, this is like spackle. It is not the blood clot. When we are talking about the blood clot, we are talking about the third step, which is coagulation, blood coagulation. Now, blood coagulation is a whole lecture by itself. I'm going to keep this on lecture exam 3. It's going to be what I'm giving you. So this is the degree of detail you need to know coagulation. But again, this is much more involved in this. The overall goal of coagulation is to get liquid fibrinogen. That's what it means by soluble. So this is the bottom line right here down at the bottom line. Remember that when we covered the components of plasma on Monday, fibrinogen was number 3 of the plasma proteins. You guys remember the first 2? Albumin, globulin. Number 3 is fibrinogen. And then you got hundreds below fibrinogen. But that's number 3 in plasma proteins. And the whole goal of this is to get this liquid fibrinogen into solid threads of fibrin. When it says insoluble, that means it's solid. And these threads of fibrin form a three-dimensional network that catches platelets and red blood cells. All right, you need to know the three pathways. The intrinsic pathway is longer. You don't need to know all the different steps to it, but it is started by activated platelets. So platelets are really important for hemostasis because it plays a role in step 2, platelet plug formation, which is not the clot, and the intrinsic pathway of step 3. Okay? And again, it's when platelets are exposed to negatively charged substances. When I do the video for Lab 12, which will be in a couple weeks, I will actually show you threads of fibrin between capillary tube glass fragments, because glass tends to form negative static electrical charge. And so if you put blood on any glass that's exposed, it'll start the coagulation sequence from the intrinsic pathway. So that's why it says collagen or glass. Now, the reason why they call that intrinsic is because all those factors in the multiple steps, like 4 steps, all those factors are intrinsic to the blood. They are found in blood. They are part of all those other plasma proteins. Again, there's hundreds of them. Okay. Then we have the one-step extrinsic pathway. It's just one step. And that means something not in the blood, and that is tissue thromboplasm. Thromboplasm is released by damaged cells. So independent of the blood, this is coming outside the blood, extrinsic pathway. And that will get to an activated factor 10. Now, what you need to know, and I think it is listed here, yes, on the second essay, where I'm going over this, it's not on a bullet here, I don't think. Maybe on the next slide it is. No, it's not. So that's the fourth bullet on essay number 2. You need to know that both of these intrinsic and extrinsic pathways meet at activated factor 10. That's where they both meet, and that begins the common pathway. Okay? Now, the common pathway is pretty short. It's right here. So I labeled this in purple, but I didn't write pathway. This is the extrinsic pathway. Extrinsic pathway, it meets the activated factor 10. The rest of this is the common pathway. Basically, all it is activated factor 10 converts inactive prothrombin to thrombin, which is an enzyme. What you do need to know, and it's listed here, is vitamin K is a precursor to prothrombin. So if you have a vitamin K deficiency, you're not going to have enough of this. If you don't have enough of this, you're not going to get very much of this. If you don't have much of this, you're not going to do the bottom line. And that's when you have a clotting disorder. Thrombin as an enzyme, that is the enzyme that does the chemical reaction that converts liquid fibrinogen into solid fibrin. And in the previous slide, that's showing you a picture from your textbook. They colored the red blood cells red, the fibrins in tan. And then the previous slide before that is showing you both the extrinsic and intrinsic pathway. Again, extrinsic pathway is faster because it's only one step. And that's what it says on the bottom of page 33, last words, extrinsic pathway, a faster shortcut to fibrin. This is because it's only one step. Okay? Now, one of the things that you should know, because this has clinical relevance, is aspirin has been found to inhibit TA2 production. Okay? And if your body can't make Ta2, it's going to inhibit the activation of platelets. Go back to the platelet plug slide. Well, because platelets, you need to activate platelets for both the platelet plug formation, step 2, and the intrinsic pathway of step 3. This is why aspirin is an effective anticoagulant. It's actually not just affecting one step, it's affecting 2. Okay? Going to this kind of fast because I got to get to immunology. This slide is the figure from your textbook, slide 37, a figure from the textbook of the common pathway. All right, now the fourth step, and this is essay number 3, is still really important. Once you have the clot and once the blood vessel walls are repaired, you got to remove the blood clot, otherwise you're going to have some problems. So when the blood vessel wall is repaired, factor 12 becomes activated, and that's going to activate inactive plasma proteins, again, one of the hundreds of the many, and it's going to activate into what we call kallikrein. Kallikrein does various things, but one of the things it does is it activates plasminogen, one of the many. Are you getting the clue that there are so many plasma proteins underneath fibrinogen? So all these are included there. Well, it gets activated in the plasmin, and what plasmin does is it actually digests fibrin, dissolving the clot. Now, hopefully, it does this very gradually and smoothly from the outside in. Every now and then, especially in larger blood vessels, if there's a large blood clot, the plasmin can actually break off pieces of the blood clot that go downstream in the blood vessel. Does anybody know what the name of that is when you get a chunk of blood clot floating in your blood? Yeah, I don't got time to wait. It's called an embolism, and hopefully you'll have other floating in the plasmin, breaking this boulder going downstream before it gets lodged into something really important like your brain or your heart. But anyway, you can see on page 56 the 4 steps in your lecture packet. 1, 2, 3, if you want to write it on page 56. Down at the very bottom is the 4th step, eliminating the blood clot. Okay. All right, we got to move on. Oh, one more thing. This is not part of the 4 steps. This is completely separate. And you only want to form blood clots when you need it. And these factors can be activated when you don't need it, and that's a real bummer. That's called a thrombosis. It's in parentheses. Forming a blood clot in an intact blood vessel is thrombosis. And that could also result in embolism, broken pieces floating downstream. So you got your liver makes antithrombin. There's also heparin that comes from the basophils, but that's just to prevent blood clots when you don't need it. When one of the exercises in lab 12C is coagulation time. And a normal range is 2 to 9 minutes. And you might think, well, you don't want to bleed very much, so the sooner the better. That's actually not true. It's actually not good if it's less than 2 minutes, because then these things on slide 39 are not working, and you are at risk for thrombosis. So it's good. It's about shades of gray. It's good to clot fast, just not too fast, because that's out of balance. Okay? All right. So page 39, we're done with the blood. Now we're on to the last topic for lecture exam 3, and that's immunology. First, let's review from anatomy, so this should go pretty fast. This should all been covered in your anatomy class. Remember that the lymphatic system is a circulatory adjunct. You have lymph vessels that are most often, not always, but most often paralleling an artery and vein. In fact, in the loose connective tissue in the body, if you've done any dissection of cadavers or animals, you can usually see little freeways, superhighways in the body of loose connective tissue where you have an artery, a deep vein, a lymphatic vessel, and a nerve. You just got these four things going throughout your body. And so this is paralleling the circulatory system. Remember that the three main functions are to transport excess fluid back to the blood. Remember, all capillaries leak. Well, you have to have a system, and it's the lymphatic system that collects all this fluid that leaks out of capillaries and brings it back to the circulatory system. And there's only two final destinations. 3/4 of your lymph, all this fluid that leaks, goes into the left subclavian vein by the thoracic duct. Only 1/4 from the right side of your head and the upper right part of your body, including the right arm, is going to drain into the right subclavian vein from the right lymphatic duct. So that's function number one. Number two has to do with, remember in anatomy, in the digestive system, you had the lacteal in the middle of the intestinal villi. Well, that's function number two, transporting most, not all, most fat that you absorb from your meals into the blood. And then main function number three is our final topic on immunology. All right, and then slide 41 is your anatomy review. Let's go over the important lymphatic structures. Remember that there are lymphoid organs or lymphatic organs. It could be said either way. Those are the ones with the fibrous capsule. So this slide 42 is about lymph nodes. You have more lymph nodes than any of the other types of organs. You have hundreds and hundreds of these throughout your body. They are filtering the lymph. So when all the lymphatic vessels are collecting all the fluid that leaks out of capillaries, the lymph nodes is filtering this fluid with a lot of phagocytes. And so if you have anything in your body that doesn't belong and it gets into lymph, hopefully your lymph nodes can localize it and get rid of it before it spreads to the rest of your body. This is also the site of lymphocyte cloning and antibody production. And you can usually feel when this is happening. When your lymph nodes are going off, making a lot of antibodies, these cells we'll look at later called plasma cells have gone through major cell cycles, IPMAT, and they're all clones. And they'll actually swell a lymph node and stretch the capsule. And when it's stretching the capsule, it's kind of tender to touch. You'll know when this is happening, when you're fighting something off. So somewhere you'll feel like, oh, that's kind of sore, and oh, yeah, I must be fighting something off. And if it does so successfully, it ends there, okay? If it doesn't end there, it can become a full body, what's called a septicemia, and then you'll have elevated body temperature. All right, the other lymphoid organ is the thymus. This is between your heart and your sternum. And this information is talking about this thymus is not always the same throughout your life. It is really big compared to the rest of your body early in life because it's really working really hard early in life to get one of the wings of your immune system going. And then after puberty, it starts to shrink, and it shrinks throughout adult life. And by the time we get these cadavers, and they're pretty old, we have difficult time finding the thymus because it is shrunk so much in a process called involution, which is on the slide underlined. In the picture on the bottom right, on the surface anyway, the first figure is showing you the relative size. Yes, it's the biggest absolute size during puberty, but then the rest of the body is bigger. So it's actually bigger relative to the rest of the size of the body in the first year of life. And when we get to talking about specific immunity, we're going to be talking about the different types of lymphocytes. And I've mentioned this briefly on Monday when I was talking about erythroblastosis fetalis, when I was talking about memory B cells and prime T cells. Those cells are all lymphocytes. We just call them B cells and T cells for short. There's also NK, natural killer lymphocytes. But there's various different types of lymphocytes. But the T lymphocytes gets its name from the thymus, because the thymus is activating those lymphocytes into becoming T cells. Okay, and that's what it's saying the second diamond. Spleen is the last lymphoid organ that I'm covering. It's the largest one, and it makes sense that it's the largest one because it is doing for the blood, this is the second diamond, it is doing for the blood what all those hundreds and hundreds of lymph nodes is doing for the lymph. Okay, you only have one spleen, but lymph nodes are filtering lymph, the fluid in the lymphatic vessels, and the spleen is filtering the blood, but you only have one. At any time, one time there's about a pint of blood in the spleen. And remember when we talked about the red blood cell life cycle, which is 4 months, about 120 days, this is one of the places where macrophages take out 4-month-old red blood cells. Macrophages in the spleen, bone marrow, and liver. Okay. Tonsils are not lymphoid organs. They're called lymphoid tissue or lymphoid nodules. There's another one that's said here. Because they lack a fibrous capsule. I made a distinction here with tonsils and MALT. MALT, M-A-L-T, stands for mucosa-associated lymphoid tissue. And all your open tubes to the outside world have MALT. And that's because there's bugs in the air and there's bugs everywhere that's trying to get into your body. And all the tubes that are open to the outside world have lymphoid tissue to hold it back. Okay? Now, tonsils are just MALT. They are actually MALT. I listed them separately because they are the first MALT for the digestive and respiratory tracts. But it's throughout the digestive system. Boom, you've got MALT. You get, we'll talk about this later, but you get into the ileum of the small intestine and you've got huge MALT, we call Peyer's patches. But even in the lungs, all that bronchial tree, MALT everywhere. You also have some MALT downstairs. All right, so the 5 tonsils, you have 2 palatine tonsils in between oral cavity and oral pharynx. You have 1 pharyngeal tonsil on the posterior roof of the nasopharynx. And then you have 2 lingual tonsils at the base of the tongue, so 5 overall. By the way, one of the earlier tests, and they're developing different types of tests for SARS-CoV-2, the virus that's causing COVID-19, it tends to accumulate in the nasopharynx. So one of the really rapid tests they were doing is we're taking a long Q-tip, shoving it straight horizontal on your nose, going right across the floor of the nasal cavity until they get to the nasopharynx, and they're just swabbing the mucus off the nasopharynx to see if they got viruses there. but that's right up against the pharyngeal tonsil. By the way, I'm going to try to weave in as much as I can with the time that I'm allotted for how this topic we're talking about now really relates to the fact that you're not in this room, because a lot of this stuff does relate. All right, so let's talk about some terminology before we get into the immune response. A pathogen, again, this is a contracted term. Pathos means disease. And so it's anything that can generate disease as a pathogen. Now, it's usually a microorganism. It doesn't necessarily have to be, but usually it is. Now, the response to pathogens are first non-specific resistance. Oh, let me move this over. Okay, so there's the lymphatic system review. But I'm going to go over non-specific resistance, and then we're going to go over specific immunity. All right, that covers it. All right. For a pathogen to cause you disease, it must first get past non-specific resistance. Until it gets past non-specific resistance, your immune system doesn't even know you've been exposed to it. Okay? It's got to get past non-specific resistance first, and if it can, now your immune system has to trigger, pick up, and take care of it. So non-specific resistance first, then specific resistance or specific immunity second. All right, so let's go over non-specific resistance. This is stuff that your body just has all the time. It's always active, or it responds with these things regardless of the virus, or regardless of even if there's microorganisms. Like in the case of inflammation, you don't even have to have microorganisms. It's going to do it. It's that non-specific. Okay, so your body just does this every time. One is species resistance, and this has to do with the fact that your parents survived to have you. And the fact that they survived to have you is because they got gifted to fight off the bugs of this world. Where do they get it? From their parents. Where do they get it? From their parents. Where do they get that? From their parents, all the way back. And the bugs of this planet, for us to survive to this day, we have had to been able to survive the bugs of this planet. If we want to project this forward from here, this is why those that have secondary conditions that cause them to be at risk for COVID-19, they don't survive this. The people that will survive this pandemic are people that tend to not have those conditions, that tend to have certain qualities so that, yeah, they'll get sick, but they'll recover, and they can recover to have children. The ones that don't, their genes die off with them. And if you do that generation after generation after generation, pretty soon you have this species resistance. Everybody understand that? If you've ever seen a movie from 15 years ago with Tom Cruise, the most recent version of War of the Worlds, I think there's a TV network that's made it a series, but this is a movie, War of the Worlds. It actually is a story written a long time ago, and there's interesting history about it. H.G. Wells wrote it. Orson Welles did a radio version of it, and people didn't hear that this was actually a drama, and his radio show of this story actually caused people to go completely bonkers and panic. So, yeah. Apparently species resistance doesn't include stupidity. All right, so, but in this Steven Spielberg version with Tom Cruise, these aliens are just, yeah, I mean, they're kicking ass. Their technology is way above ours. We just look like little ants. Looks like all is lost. And then the aliens catch the cold. And our cold, which, because we have species resistance and the common cold, we can just, yeah, it's annoying, but we survive it, kills them all. They don't have species resistance. And if you were wondering why I had pictures in the middle of the slide about War of the Worlds with Tom Cruise, that whole story, if you watch that movie, you actually see it. You know, all of a sudden they got runny noses and they just die from the cold. All right, mechanical barriers. And this is just common sense: intact skin, intact mucous membranes. The picture that is on the top right is of a spirochete bacteria, the same one that causes syphilis. So yeah, and that's what PPE is for. PPE is to accentuate mechanical barriers. And don't even get me started talking about PPE and this fiasco, because there was no excuse for having this kind of shortage. And then we have enzyme activities. They're in the fluids that are exposed to the outside world, and you can see them listed there: tears, saliva, stomach fluids, the mucus from goblet cells in your respiratory tract. All those have various proteins and enzymes that make things tough for microbes. It's not 100% foolproof, it's not 100% effective, but it contributes to species resistance. Those that have more of these non-specific enzymes tend to survive to have kids. The ones that don't tend to die off. So more can survive to have kids that have enzymes. Eventually our species have these enzymes. Not only are they enzymes, it's also, we'll look at later, specific antibody, IgA, that's also put in exocrine fluids. It's very non-specific, but it is interfering with infective microbes. Also complement proteins, also punch holes in bacterial membranes. And that's the picture on the bottom right of the slide. It's showing you how these proteins act together in a coordinated way to actually punch physical holes in bacteria. Okay. Non-specific though, it'll do it for any bacteria, non-specific. And then we have interferon. If you have the PowerPoint going, this is a good time to actually follow the animation on the bottom left. But this also adds the species resistance. If you watch this animations, and definitely watch, especially since we're not, in the whale room, it's really good to download the PowerPoint and actually watch the animations because it explains a lot. Okay. I don't have time to wait for the animations, but this is the story. When a virus, again, viruses are not living, but because they are biochemicals, they can hijack a cell into becoming viral-making factories. What some cells, and especially if you have good species resistance, your cells will make interferon. And probably the best analogy for the interferon is the cell that is first infected becomes the soldier, the Medal of Honor winner, because he jumps on the grenade and takes one for the team so that the other soldiers, his buddies, survive. So once a cell gets infected with a virus, before it becomes a total virus-making factory, and this is what's in this animation, it will release these interferon proteins. Now, this cell that's releasing the interferon proteins is on the grenade. It's going to die after it makes thousands of viruses. It's dead. But before it dies, it releases interferon. Interferon will go to the adjacent cells that have interferon receptors. And when interferon binds to the receptors, it causes that cell to make a bunch of antiviral proteins. And so when the virus comes to infect those other cells and turn those into viral-making factories, they can't because of interferon-activated antiviral proteins from the first cell who died, but saved all his buddies. That's interferon. Okay, then we got inflammation and fever. Hopefully this was discussed fully in your anatomy class. I know I cover it. It's basically chemical-mediated response. The chemical is histamine. Histamine comes mostly from tissue mast cells. If there are any basophils in the area, they will also do it, but remember, basophils are the least common white blood cells, so chances are there won't be any basophils there. The histamine causes vasodilation. That causes everything to get red and warm. And when you get vasodilation, you're going from a blood vessel of that diameter, this is a cross section, to a blood vessel of this diameter, vasodilation. But these endothelial cells are not growing or stretching, they're separating. And because of that, blood vessels become very leaky. And this is all described on the slide. And a bunch of these good things that you want to go get out to the tissues to fight off infections or other things are able to get out. Okay? But when a bunch of fluid leaks out of this, that's why you also get swelling. And histamine also lowers the threshold of nociceptors. You guys remember nociceptors? And so in clinical courses, you will hear the 4 pillars of inflammation, which I wrote in red. Redness, warm, swelling, and pain. The last arrow on the bottom of this slide on inflammation, slide 50, is talking about the phagocytes. Fast-acting white blood cells are neutrophils. Hopefully, yesterday down there on the whiteboard, you wrote fast eaters. Neutrophils are the fast eaters. They're there first. But after about a week, they get full, and that's when the macrophages start coming in big numbers, and those are the big eaters. Remember, they're monocytes here, macrophages out here. And they don't get full, they just keep eating, okay? Now let's expand on this last point of phagocytosis. So this is actually under inflammation, because it's the last arrow on this slide. Inflammatory phagocytosis by the fast-acting neutrophils and macrophages. This figure is showing you the classic sequence on page 51. But then on page 50, slide 52 is this inflammatory phagocytosis. Again, neutrophils come first, monocyte macrophages come second. How do they know where to go? The way they know where to go is because of a process called chemotaxis. Oh, but first, let's differentiate between those that are free, able to move around, and that's the neutrophils, monocyte macrophages, and those that are fixed. Your body has, in various parts of your body, fixed phagocytes, and that's like the reticuloendothelial cells in the liver, alveolar macrophages in your lung alveoli, microglia in the CNS, one of the neuroglial cells. So those are all fixed. They're not moving around, but the white blood cells, neutrophils, monocytes, those are moving around. And it's those ones that have to go through this chemotaxis, and there's two sub-kinds that are written on the bottom of this slide, 52. A positive chemotaxis is an attraction. Negative chemotaxis is a repulsion. Everybody see that on the bottom of this slide? So this may start out random. You know, the injury just happens to be where there's some neutrophils. They start going toward it. Where it actually happens is kind of random. Once it gets going, chemotaxis makes it decidedly not random. It is now guided by chemicals. Okay? Slide 53 is showing you a textbook picture from your textbook on phagocytosis. I think the really interesting thing about this figure is that it shows that if you trip and fall and scrape your knee, you're going to get some white blood cells eating bacteria, because everybody's skin is covered with bacteria. You scrape your knee, some goes in your body. Those phagocytes will actually rip apart that bacteria into the 4 biologically important macromolecules. The digestive enzymes in the lysosomes will break all those into single link amino acids and glucose, and your phagocyte will actually make ATP from bacteria and viruses that it makes. So when you think that you feed all your cells, yeah, for the most part, but every now and then, if you get something in your body that don't belong, that is biological, your phagocyte will actually make ATP out of it. One other thing too is macrophages, you might hear the term dendritic cell. Dendritic cell is basically a type of macrophage that throws out really long foot processes or arms out to grab bacteria or other things and brings it back in. And when you have a whole bunch of arms going out of the cell bodies of these macrophages, it almost looks like a neuron cell body with dendrites. That's why they call it dendritic cell. So if you ever wonder why they call some macrophages dendritic cells, that's why. All right, so that's non-specific resistance. Everything that's going to be handled by your specific immune response must get past non-specific resistance. Let's go back to Monday's lecture, and we talked about, and some of these concepts, you want to tie this back to erythroblastosis fetalis because they are connected. The reason why the loading dose for a primary immune response is so much bigger, you got to take the whole vial of flu vaccine, is because that flu vaccine has got to get past this in order for this to make antibodies for that flu bug. That's why you have to have a much bigger loading dose for a primary immune response. So some of this ties together. Hopefully I'll have time to tie some of this together. All right. We are on slide 54, where we're talking about specific immunity, specific resistance. This means your body's immune system is not only going to be individually responsive to this one, say, flu virus, but it's so specific that handling this year's flu virus may not work next year and probably won't, which is why you have to get a flu shot every year. Right now, SARS-CoV-2, it is unclear whether or not this is going to be a seasonal bug like the flu virus. If it is, then those that catch this version and recover may not have lifelong immunity for this COVID-19 bug. We may have to get annual vaccines, just like the flu. The jury's still out on that. There are some signs that point one way and some signs that point the other way. It would be really nice if it didn't mutate and we just had one version, and once you got it, you are assured to be immune for it for the rest of your life. But right now, probably not. All right. What I'm going to do is I'm going to cover the basics of specific immunity, some basic principles. I'm going to talk about it in different ways. A lot of it is the same thing, but in different ways. A lot of it is interconnected, kind of like that's why the loading dose for the primary immune response has to be much higher, because it's got to get past this to get to that specific immunity. I'm going to talk about the main cells that are involved in specific immunity, and then I'm going to get into the details. So first, big picture things, and then we'll get into the details. Now, there's a lot more to this topic than I'm giving you, okay? When I was at UCLA, I had 2 quarters of immunology. I'm giving you 2 lectures. My anatomy class gets 2 PowerPoint slides. But you only get 2 lectures. There's a lot to it. Just concentrate on the degree of detail I'm giving you, because that's what you're going to be responsible for. All right. Second bullet on this slide is for maximum effectiveness, you're going to need to have a previous exposure. This is the norm. This is like the flu shot. This is like the RH system in blood typing. That is like the usual. And you need to write this down on blood typing from Monday's lecture, where I'm talking about the ABO system. It says it, it's highlighted, but you better really write it down next to that slide. ABO is the exception. You do not have to be exposed to the other blood types to have the antibodies. You are born with the antibodies regardless of your exposure. That is the freak exception. Everything else follows this second bullet. You have to have a previous exposure. The best way to conceptualize specific immunity is it's got 2 forms, and that's on the very bottom. So it's like 2 wings of a bird. You have cell-mediated immunity and antibody-mediated immunity. Both together is specific immunity. You need both. This antibody-mediated immunity is also referred to as humoral immunity. This means the same thing. Now this works for all pathogens that get past non-specific resistance. But the best way to understand it is how the specific immunity responds to viruses. And it's because viruses turn your cells into viral-making factories. Now, I'll just give you this as a backdrop to this whole thing. But antibodies can take out free-floating viruses in your body, but the cell-mediated wing takes out your cells that have become viral-making factories. That's why it's called cell-mediated. Okay? All right. Slide 55 is a slide from your textbook showing you cell-mediated wing. It's taking out your cells. So down at the bottom right of that figure is your cell that's become a viral-making factory, and the cell-mediated wing kills your cell. Okay? And then on the left side, you can see the plasma cells secreting antibodies, and antibodies can attack free-floating viruses. Then I included a figure from the fourth edition because I really liked it. I don't know why they changed it, maybe so that they can make more money off of you students. All right, so let's talk about the basics here, starting on slide 57. Remember that an antigen is anything that causes your immune system to make antibodies. Antigen means antibody generating. An antibody is an immunoglobulin. Now, not all immunoglobulins are floating in plasma, but the ones that are contributing to globulins being the second out of all the plasma proteins. Albumin, globulin, fibrinogen, and then many others. Okay, but antigen is anything that causes your immune system to generate antibodies. It doesn't even have to be a microbe. Your body will make antibodies to chemicals. That chemical is an antigen. And much of what, not 100%, but much of what this is doing, it's doing through binding antibodies to antigens. And when the antibody binds to an antigen, it's called an antibody-antigen complex. Now, if you look at this figure on slide 57, you'll see that antibodies look like capital Y's, and it's the upper arms of the capital Y's that binds to antigens. Okay. And on slide 58 from your textbook, you could see that the upper arms of the Y's of the antibodies are actually binding to these things called epitopes on the antigen. So it doesn't bind to the whole antigen, it binds to epitopes on an antigen. We'll talk about epitopes more a little later. All right, now let's talk about the main cells involved. And these are mostly lymphocytes. Remember that T cells became T cells because of the thymus. In fact, the thymus is also referred to as an endocrine gland because it releases a hormone called thymosin, which causes lymphocytes to differentiate into T cells. Okay. Now, T cells represent 50% of all lymphocytes, but most of them that circulate in the blood and the lymph are T cells. A lot higher percentage are T cells because they're moving around about your body looking to take viral infected cells of yours out, and you got to move around to do that. So remember I said when we were covering on Monday the 5 different white blood cells in your pipes, lymphocytes are number 2, but throughout your whole body, lymphocytes are number 1, because all the lymphocytes in the lymph, all the lymphocytes in all your lymph nodes and in the spleen, they're not counted in the white blood cells that are circulating. You have a whole bunch of lymphocytes that aren't circulating in blood. But the ones that are circulating in blood, over 3/4 of them are T cells. Now there are 3 different types of T cells, and that's on the next slide, 60. Okay. The one that actually does the killing is on top of the first diamond, the effector cytotoxic T cell, and it kills your cells that have become viral-making factories by touch killing. It actually has to bind on a receptor. It actually has to touch the infected cell to kill it. Now I'm introducing this as the cell that kills your cells that have become viral-making factories, but these are also the cells that kill cancer cells. They'll recognize cancer cells in your body as being foreign, kill them out. They are also if you need a tissue transplant, because you have different self-markers on the transplanted tissue. They have to give you immunosuppressive drugs to kind of suppress cytotoxic T cells, because these are the cells that will reject tissue transplants. Then we have the second one is primed T cells or memory T cells. I mentioned this on erythroblastosis fetalis. This is one of the cells you don't want to have, and that's what you're using RhoGAM for, is you don't want to have primed T cells to make anti-D. And then the last one is the helper T cells. And this is key. If these two are 2 wings of a bird, the helper T cell stimulates both wings. It is the only cell that is in both wings. Okay, this will be relevant when we talk about HIV. Helper T cells, and on slide 61 is showing you some of the chemicals that helper T cells release to stimulate both wings. That's from your textbook, slide 61. All right, slide 62 is the B cells, and there are only 2 B cells. The effector B cell is also referred to as the plasma cell. I know it's kind of a weird name, but this is the one that secretes antibodies. And then memory B cells are held in reserve in case you catch that bug again, in case you get the flu vaccine. That is to give you memory B cells for that year's flu, so that if somebody sneezes in the elevator, you're going to generate a bunch of antibodies for that flu bug in a hurry because you have memory B cells. But obviously this is going to be intermingled here. I'm going to go over this in a little bit more detail. But the killing cell for the cell-mediated wing is the effector cytotoxic T cells. And the killing cell for the antibody-mediated wing, since it secretes antibodies, is the plasma cell. And slide 63 is from your textbook showing you both the memory B cells and the plasma cells. Plasma cells actually get huge because they are secreting so many proteins. Again, these antibodies are proteins. Huge amounts of rough endoplasmic reticulum when they're making antibodies. All right, now this slide 64, let's go back here. On the very first slide on specific immunity with that bird on the bottom of the slide because of the two wings. By the way, do you guys know what bird that is? What slide is that? 54. Slide 54. It's not a seagull. On the bottom. That's an albatross. Albatross looks like a seagull, except its wingspan is 6 to 8 feet. But anyway, if you look at that slide, and it says, for maximum effectiveness, a previous exposure to a foreign agent or pathogen is generally required. The second bullet on page 54 is totally tied to slide 64. I'm talking about the same thing on slide 64, and this is immune memory. Okay, now I talked about immune memory, and also if you have your lecture exam 3 essays, go to number 4 where we talk about erythroblastosis fetalis, because these are the terms that I kind of had to talk about when I was going over that on Monday. Now, if you look at slide 64, you will see the first lump in blue. Let's go to either the slide, the figure on the bottom right, or the one figure on the bottom left. That first lump on the left is the primary immune response. Either circle those words there or write it. The first lump on the left, primary immune response. Notice it is wimpy and slow. But then after the primary immune response, you get memory B cells and prime T cells. Now look at the second lump. It is much steeper, which means it's much faster, and it's much bigger. That's the secondary immune response. That is tied to 10 slides ago when it says, for maximum effectiveness, you generally need a primary immune response. A previous exposure to get you a primary immune response. Then, if you're exposed again, you would get this fast, huge lump. Stronger and faster. Later on, I'm going to talk about the clonal selection theory. The clonal selection theory is not another topic. It's just explaining this in more detail. Okay? Immune memory. So, now look at that essay number 4. you're given anti-D from another source. The key here is the mom did not make the anti-D. A cow made the anti-D. And we'll talk about different forms of immunity, and we'll tie this to passive artificial immunity later. Because the trick here is to take out the D antigens, which are agglutinogens, this is the third bullet, before the mom's immune system generates this first wimpy lump. Because if the mom doesn't make that first wimpy lump, the mom does not have prime T cells or memory B cells. And if you have no memory B cells, every pregnancy is immunologically like the first. And remember, the first Rh positive baby is separated from the placenta, has no problem. And if each pregnancy can be like that, an Rh negative woman can have multiple Rh positive births and never have an issue. Okay? Does that help at all? Tying that to this slide 64 and those lumps? If a woman doesn't get RhoGAM and has memory B cells and prime T cells, now those few Rh positive fetal blood cells that cross the placenta, because a few cross, is enough of a loading dose to activate these guys. And they will make this huge, fast anti-D titer, which would be the second lump. And that much anti-D crosses the placenta and agglutinates fetal blood in utero. Slide 65 is showing you the same thing, same type of graph, but separated them out as a primary response and secondary response. Just notice that the secondary response is bigger and faster. All right, then on slide 66, I'm doing pretty good on time, is an overall figure. And I really like this figure because it covers a lot of concepts in one figure. And I really like big picture things like this, instead of breaking it up into different fragmented figures and then you got to put it all together. This puts it all together. Now, if you look at this figure on this slide, well, with the red with all the cells, where is cell-mediated immunity? It's on the top. Where's antibody-mediated immunity? It's on the bottom with the plasma cells secreting antibodies. Look right in the middle. What is the cell that is in both wings? The helper T cell. Now this figure, I have separated things out to slide 67 in this table. And slide 67 is copied and pasted as essay number 5 on your essay handout. That's it. I've just covered all the essays for lecture exam 3. But let's take a few minutes to go over this. First, let's look at the cell-mediated column. Here you're involving all 3 T cells. Those are the cells involved, but the actual killing cell, as I pointed out in the blue arrow, is the cytotoxic T cell. And it kills by touch killing your cells. What is it targeting? Viruses inside your cells that have turned your cells into viral-making factories. But it will also take out organ transplants and cancer cells. Antibody-mediated wing. It has both B cells, effector B cells and memory T cells, plus helper T cells. Helper T cells are the only lymphocytes in both wings. The actual killing cell is the plasma cell, and it does so by secreting antibodies. Now, here I'm giving you a slight introduction to the different types of immunoglobulins, antibodies. The most common is IgG. IgA is what I told you is made in tears and spit and fluids that are exposed to the outside world. Just a preview of coming attractions. Right now they're talking about SARS-CoV-2 testing, and especially testing by antibodies, which are actually showing you what kind of, maybe you caught it, didn't know it, and now you're immune to it. Well, that's, you got to have the antibodies. And there's an IgM, which I told you is the anti-A, anti-B, anti-D are all IgMs. Well, when your body first gets infected by SARS-CoV-2, you make a lot of IgMs for it. And if you have in the antibody blood test a lot of IgMs, you still got to quarantine because you're still fighting this thing off. You still got viruses in you that the IgM is trying to help take out. If you have a lot of the IgG, and that's on the slide, for SARS-CoV-2, chances are you are immune. You've already beaten it. Now these are the immunoglobulins of reserve in case you get infected with that again. All right, so antibodies are targeting free-floating viruses, extracellular pathogens. These are also the antibodies that are attacking various chemicals. And the antibodies, again, is what's doing the killing or inactivating. So when you have time, check this essay out, hold it against that figure on the previous slide, and see if you can't see all the things in that table on that one figure, which is why I like that figure. Slide 68 is showing you a picture from your textbook of how the cytotoxic T cell has to bind to an infected cell to touch kill it. It actually does have to touch that cell. That's why it's called touch kill. And then slide 69 is showing you the antibody-mediated wing, again with plasma cells secreting the plasma. The last slide of the PowerPoint, again, you got to add 7 days to that. Lecture exam 3 will be on April 29th. Why don't I go ahead and do a close-up of the whiteboard. While I'm doing that, if you have any questions, now's the time to ask, because I actually got through 40 slides today. Yeah, working my ass off. Okay, this is the left bank, showing you the 4 steps of hemostasis. All right. This is the middle bank, showing you the anatomy review of the lymphatic system. And the first part of the immune response, which is non-specific resistance. Again, to activate specific immunity, you first got to get past non-specific resistance, which, by the way, is the reason why HIV has not been the terrible plague that it could have been. And that is, HIV actually has a difficult time getting past non-specific resistance compared to other bugs like SARS-CoV-2. HIV is, you have to get a decent amount of it to actually become, to test positive for HIV. Man, that's good news. I mean, nobody should be handling needles anymore, but a lot of the ways that healthcare workers have changed the way we handle needles came from HIV, and just the panic that it generated when people would have needle stick accidents. Well, because HIV has a hard time getting past nonspecific resistance, only 1/10 of 1% of healthcare workers who had needle stick accidents from the 80s and 90s actually seroconverted to being HIV positive. And that's because HIV has a hard time getting past nonspecific resistance. But do you see how I drew the blue line from thymus to the T cells? That is the connection there. And then this is all I wrote here for specific immunity. There'll be more on Monday. Monday will be the last lecture for lecture exam 3. Okay? Any questions? Again, on the last slide, add 7 days to that. Lecture exam 3 will be 4/29. All right? If there are no questions, I'm signing off.